Here is my paper. It's titled All About Hexokinase. Proteins are the main building blocks from which cells are assembled. Proteins have many different functions in life. Just to name a few, proteins can catalyze reactions, provide structure to tissues, provide transportation, receive information, store amino acids, etc. Proteins are very complex molecules. This is partly why they have 4 different levels of structure. As discussed in lecture, the primary structure has a unique amino acid sequence having about 20 amino acids. There is great diversity due to the mutations in DNA, and they are formed by peptide bonds. DNA transcribes into RNA, which translates into protein to make this structure. The primary structure is a chain with no branching. The secondary structure is made up of regular repeated folding of the polypeptide chain. This can be in an alpha helix, which are coils, or beta sheets, which are just folds. Tertiary structures are 3-dimensional, formed by an entire polypeptide chain, including both alpha helices and beta pleated sheets that lay on top of each other. This can be made up of the 5 different bonds in chemistry, which are disulfide bonds, ionic bonds, hydrogen bonding, hydrophobic interactions, or van der Waals attractions. And lastly, there are quaternary structures that are made up of more than one polypeptide chain. They are even broken up into different domains due to their complexity, and are usually made up of 100 to 250 amino acids. The structure of proteins are vital to their function, because without the proper structure, they will not work properly. This is the general overview of proteins. Now let me discuss a specific protein, hexokinase. Hexokinase is a protein involved in the initial step of glycolysis, or the splitting of sugars. During this first step, glucose forms a sugar phosphate after being phosphorylated by adenosine triphosphate, or ATP. The phosphate will then hold a negative charge, which keeps the sugar from passing through the plasma membrane, so it remains locked in the cell. This reaction goes as follows. Glucose plus ATP forms glucose 6-phosphate and ADP and hydrogen. In this reaction, hexokinase works as a catalyst in the reaction to form a hexose phosphate, which in this case is glucose 6-phosphate. The function of hexokinase can be inferred from its etymology. Hexo, meaning 6, represents the hexoses, or 6-carbon sugars, that hexokinase phosphorylates. The kinase stands for the movement, as in the study of kinesiology, which you can think of as the movement of a phosphate group from one molecule to another molecule. Kinase in biology also specifically refers to an enzyme catalyzing a reaction, transferring energy from a donor, usually in the form of ATP, to an acceptor. Many people make the mistake of confusing hexokinase with glucokinase because of the fact that hexokinase usually phosphorylates glucose and they have similar functions. These 2 enzymes are not the same, however, so they should not be used interchangeably. Glucokinase is an enzyme that also phosphorylates glucose to form glucose 6-phosphate. Glucokinase is only present in the liver cells, while hexokinase is present in all cells, which is why glucokinase is also known as hexokinase 4. Mutations in glucokinase relate to diabetes, hyper and hypoglycemia. Hexokinase is a structural protein that also has 3 other isoforms pertaining to different cells. Hexokinase 1, or HK1, is found in most tissues, usually the brain or red blood cells, because energy production is critical there. HK2 is found in muscle tissues or areas that are insulin sensitive, and both HK1 and HK2 are abundant throughout organisms. HK3 is found in human leukocytes, and HK4, glucokinase, previously mentioned, is very similar in function. Both HK3 and HK4, unlike HK1 and HK2, are cytoplasmic due to the fact that they lack a mitochondrial binding locus. Also, native HK1 and HK2 have the ability to bind to the outer mitochondrial membrane, which induces oligomerization. Hexokinase is an important substrate because it is found in all organisms in one form or another. The structure of hexokinase has been solved for its tertiary structure, and other structures have yet to be fully solved. As you can see, the enzyme is made up of many alpha helices and beta sheets, and the tertiary structure and the N and C terminals are very similar to each other. This has been determined for human HK1 and HK4, but many other structures within the HK family are still being hypothesized. The isozymes also have different properties making up their structure, which makes it difficult to pinpoint hexokinase structures. HK3, for example, lacks the hydrophobic N-terminal sequence, critical for targeting to mitochondria. There is quite a bit of information in the scientific community regarding the research of hexokinase since it is relative to a multitude of human bodily functions. However, not all research agrees, and throughout the past 50-odd years, researchers are still searching for answers to questions on hexokinase. The functioning of hexokinase is composed of several subunits that fit together. After the first step of the metabolism of glucose, once hexokinase has created a 6-carbon sugar, that glucose 6-phosphate has several options of pathways to enter. The 4 isozymes of hexokinase differ in their subcellular localization and other properties which are important when determining the pattern of glycolysis in cells. Therefore, you could divide the function of hexokinase into the subunits of the 4 different isozymes that exist since they pertain to different tissues in the body. The genetic information regarding hexokinase is a topic of high interest in the medical community. There are several genes that code for hexokinase because hexokinase is so diverse. Hexokinase is found in many different organisms and many different parts of organisms. Most of the information and research has been found on the human genome and other animals that are used in scientific study, like chimpanzees and lab rats. The coding sequence for the HK1 gene has been found in one scientific study and shown in the following table regarding different primers that were used. Research done on lab rats and mice have found many different discoveries in the genetic diseases associated with hexokinase. There are many theories on how HK1, 2, 3, or 4 relate to different diseases. Since hexokinase is a catalyst in energy production, its technical difficulties create mutations in genes. Hexokinase has been used to determine hepatoma in rats, which can be correlated to human genes. With the breakthroughs in today's medicine, people are now living longer and cancer is becoming more prevalent. The emergence of cancer in the community has forced researchers to try to understand cancer cells and search for a cure. Hexokinase 2 is related to tumor cells and cancer in organisms. Research has been done on mice reflecting that the reduction of HK2 can inhibit the growth of tumor cells in mice. These are just a few of the genetic diseases that are related to hexokinase and glycolysis. I also previously mentioned diabetes and hypoglycemia. Hopefully, future research will give us more insight into how hexokinase in the body can affect these diseases so that more people will live healthily. Hexokinase is a complex protein made up of 4 different isozymes. The function of hexokinase is to regulate energy in the cell, so it is vital to all living organisms, including you. Therefore, the understanding of the function, structure, and capabilities of this enzyme can better the medical community. In a sense, proteins make the world go round.